Hey CW Bio, Mr. Kennedy here. Today, we're going to talk a little bit about chapter 12 of your book, The Rise of Genetics, or basically the history of the discovery of DNA, which led to, well, genetics. So if we go back in time and we revisit Mr. Kennedy's childhood, we need to take a look at some historical events that began on or about the year 1869. Going way back into the 1800s, we enter a time period where people had no clue what DNA is, what it looks like, what it can do. Those are things that we kind of take for granted in our day-to-day lives because, well, I don't know about you, but ever since I was a little kid, people have been talking about DNA in some form or another. DNA was discovered by a man by the name of Frederick Meisner. He was a physician who basically used an enzyme called protease to digest proteins away from white blood cells in blood samples that he had taken to study. Ultimately, as he mixed this material around, he found that the protease enzyme digested pretty much everything in his sample. But when he pulled this little, stir stick out, he found this material clinging from it that, well, he could only describe as like a stringy ball of pus. I know it's gross, but here's a guy digesting away proteins in his cell sample, and you would expect there to be nothing left in there after you stir it around for a while. He found this gelatinous pus-like material that, well, he had no clue what it was for or what it could do. In 1869, we didn't have like electron microscopes and all kinds of fancy lab equipment to try to explore what this material was. About all he could do was take a pH measurement of it where he found that it was acidic. He hypothesized that since he had used a protease enzyme to digest all the material of the cell away as he mixed it around, that maybe this material, that stringy ball of pus that he was left with, came from the nucleus of a cell. So he put two and two together and named this stringy ball of pus nucleic acid. Sound familiar? That's the Na in DNA, right? Nucleic acid, deoxyribonucleic acid. Meisner didn't know it, 1869, but he was onto something. So if we fast forward from 1869 to about 1928, we are introduced to another Frederick, this one Griffith, who in 1928 was working with pneumonia-causing bacteria, or basically pneumococcus. There are two different strains of pneumococcus even in 1928, and one is deadly, also known as the virulent strain, and the other is not deadly, also known as the non-virulent strain. So this guy, Griffith, in 1928 is playing around with some pretty scary stuff because realistically, if you get pneumonia in 1928, there's no way for us to save your bacon. There's no antibiotics or anything like that. You'll just have to fight it off yourself, and if you can't, you might die. So this was a pretty brave experimental process that Griffith embarked upon in 1928. Let's look at what he did. So here's the virulent form of the pneumococcus bacteria that he studied. The virulent form, again, is lethal. It causes pneumonia. It'll make you really sick. When you have pneumonia, your lungs basically fill up with fluid and you can't breathe from the infection that's going on inside of there. One of the most notable things that distinguishes this virulent form of pneumococcus bacteria from other pneumococcus bacteria is it has a smooth outer coat to it, which makes it hard for your immune system to recognize. Therefore, as this bacteria grows in your body, your immune system really struggles to find it and kill it. That means the bacteria can reproduce pretty quickly and make you really, really sick. Sometimes this virulent form is just referred to as the smooth form of pneumococcus bacteria because of that smooth outer coat. Next is the harmless form. The harmless form, well, I didn't put a picture on here because really you'd have to have a microscope, an electron microscope, to see the difference between this and the smooth. But at the end of the day, the harmless form is harmless. because it doesn't have a smooth outer coat. It has a rough, bumpy outer coat that is actually pretty easy for your immune system to recognize. You could have harmless pneumococcus bacteria in your body right now, and it's no big deal. You're not going to get sick because your body recognizes the bacteria, can gobble it up and get rid of it before it has a chance to really make you ill. So the harmless is sometimes called harmless, it's sometimes called non-virulent, and sometimes just rough outer coat. Well, here's what Griffith did. Let's check this out. Now, if you're a fan of rats and mice, you're probably not going to be a fan of Griffith because basically he performed some experiments on rats and mice to, well, basically study the different types of pneumococcus bacteria in vivo. That means in a living system. So basically what he did is he took the rough-strained pneumococcus bacteria and injected that into a mouse. That's what you see going on here. If you take the rough strain, inject it into a mouse, and you know that the rough strain is not harmless, then the end result there is something that you probably expect. The rough pneumococcus bacteria did not hurt the mouse, okay, or rat, all right? It lived on just fine. Okay, so that kind of establishes that non-virulent or rough pneumococcus bacteria is indeed harmless. And then he went to the next level where he wanted to basically prove that harmful is harmful. So he takes the smooth-coated pneumococcus bacteria, puts that into a mouse, and lo, all the mouse died. Dun, 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 right? Like, I mean, yeah, like it's harmful, so yeah, they died. Then we get to the true experimental stage of what he did. He took the living smooth strain and boiled it, okay? Basically heated it up for like 20 minutes, in essence killing all of the smooth strain pneumococcus bacteria, injected that into a mouse, and as luck would have it, all the mice survived. Kind of makes sense, right? So here we've established the rough, non-virulent bacteria is indeed not harmful, and that living virulent bacteria is indeed dangerous. And if I heat kill my living, like dangerous bacteria, then I can make it not dangerous and the mice all survive. So here's the weird part. In the last experiment, if you look closely there on that slide, you can see that Griffith actually mixed two things. He mixed his living rough strain pneumococcus bacteria, which is supposed to be non-virulent, not harmful, with his heat-killed smooth strain, which again is supposed to be not harmful, not deadly for his mice. Mix that, put it in the mouse, and lo, all the mice died. Like, what's up with that? Like, I put two things in a mouse that when injected by themselves did not hurt the mouse. How come when I put them together in the mouse dies? So Griffith does this little Minnie Mouse autopsy in 1928. And again, we're kind of limited on what kind of technology we have available to research the end result. The one thing that he did find, which was really, really weird for him, was that he didn't find any dead, rough pneumococcus bacteria in his now dead mice. He also did not find any dead or heat-killed smooth. He basically found that his dead mice were riddled full of living, smooth-coated, virulent bacteria, which for him was really, really weird because he didn't put that in there. Like, how did that happen? How did he get live, like, virulent bacteria in those mice when he didn't inject that? Well, it's 1928, and poor Griffith had no clue. So just to kind of give you what he did from this picture in words, let me click through a few slides for you here so you can jot this down. So we got live cells, harmless. First, he took the live cells, injected them into the mice. Mice lived, okay? Then he took the live virulent cells, injected that into the mice. Mice died, okay? We know that from that picture. And then he takes the heat-killed virulent cells, injects that into the mice. Mice lived. And lastly, he mixes the live heat-killed the live harmless and the heat killed virulent, and all the mice died. Okay, so here's basically his results and where his experiment ended. He did his Minnie Mouse autopsy, like I said, found the living, smooth, or heat-killed virulent cells in there, and concluded that, well, somehow the harmless bacteria must have been transformed into the deadly stuff. He had no idea how, but somehow it happened. So he just said there was some sort of transformation principle that caused this transformation to happen. He was unable in 1928 to figure out what that was. So we got to fast forward about a decade to 1941 and a scientist by the name of Avery. Avery is super important because Avery is the first person to identify Griffith's transformation principle. Okay. So I'm just going to sum up what Avery did by telling you this. He basically repeated Griffith's experiments with the pneumococcus bacteria with one minor tweak. He didn't mix whole heat-killed virulent cells with his live harmless cells like Griffith did in his final experiment. No. He basically heat-killed his virulent cells and then kind of ran them through this machine called a centrifuge to separate out the different building blocks or components of those heat-killed cells. He separated out the lipids from the rest of the cell, the proteins from the rest of those heat-killed cells, the nucleic acids and the carbohydrates. He separated each major category of biomolecule one from the next, and then he would mix those with live harmless cells and inject that into a mouse, okay? And guess what he found out? Come on, I know you really, really want to guess, right? Well, long story short, the only thing that caused transformation was when he mixed nucleic acids from the heat-killed, smooth, or virulent pneumococcus bacteria with the live harmless. That was the only time he saw a transformation. So here we are, 1941, and basically this is like World War II time. I mean, we have cars, we have airplanes, right? It's modern America, and we've just discovered that there is this molecule in a cell, well, basically in the nucleus of the cell, remember nucleic acid, that can cause some sort of transformation to occur or control what we look like at a cellular level. But we still have no idea how it does it. We don't even know what DNA looks like yet, right? And it's 1941. Pretty interesting stuff. Now, here's the summary of what Griffith did. So again, what Avery did. Again, this is what, I just explained a moment ago as kind of a repeat of Griffith's experiments. So Avery's experiments were similar to Griffith's with some minor changes. You can see there he took the virulent strain of bacteria, broke it down into its individual parts, mixed them one at a time, proteins, carbs, lipids, and DNA with harmless bacteria, injected that into the mice, and only when he mixed the nucleic acid with the harmless bacteria did he get a transformation. Okay, so DNA at this point, we know it lives in the nucleus of a cell and it can cause a transformation. That's all we know. Fast forward to 1952. It's taken another decade and entered two new scientists, Hershey and Chase. No, this is not the candy bar guy. Okay, so Hershey and Chase, I don't want to belabor the point here, but basically they studied viruses, and their whole idea was to try to figure out how DNA maybe controlled what was going on in a cell. Does it do it from outside the cell? Does it do it from inside the cell? So they radioactively labeled some virus particles. Basically, they labeled the protein code of a virus with one type of radioactive label, and then the DNA inside the virus with a different kind of radioactive label. And cool thing is they basically used an X-ray machine to take pictures of a virus as it infected a cell. And what they found out was that, well, in order for DNA to do anything to a cell, take over, change it, control it in whatever aspect you want to consider it, the DNA has to get inside the cell. So they literally watched a virus using an X-ray machine inject DNA into a cell and then that DNA take over the cell. like, whoa, that's pretty cool in 1952. Okay, if you're a fan of movies, 1952 is right about the time, Marty flew the time machine back to the future and, had that whole issue in Hill Valley with lightning striking a clock. Back to the Future movie. Anyways, so here's kind of a summary of what they did. They wanted to see how the DNA could get into the cell. They used radioactively labeled bacteriophage DNA, mixed it with E. coli. Through the use of X-rays, they were able to photograph the DNA moving from the virus to the bacteria. Still don't know what DNA looks like, though. It's 1952. All right, so we're going to fast forward now to two fellas by the names of Watson and Crick. James Watson and his buddy Francis Crick. Okay, so Watson and Crick. So these guys are given credit for developing the structure of DNA. Interestingly enough, they didn't do a whole lot of their own research, but they were very good at collecting data from other scientists, and they used that data to kind of put things together in the model that we now refer to as the double helix. Okay, so Watson and Crick, they obtained information from a handful of other folks. This is that list. Basically, a guy by the name of Irwin Schwartz, Linus Pauling, and then two contemporaries that were literally competing with Watson and Crick for discovering the DNA structure, Rosalind Franklin and Maurice Wilkins. So what did these people give them? How did it help? We'll start with Schwartz. Irwin Schwartz was a biochemist, and he had been studying DNA for years. It's Irwin Schwartz who basically explained, through his research that every, A has to pair to a T and a G has to pair with the C in terms of DNA. So Schwartz is our guy that we give credit for complementary base pairing in all the DNA that he studied. There were always equal proportions of A and T and G and C. So he concluded that A must be bound to T, T to A, and G to C, and C to G. Okay, so that's what Schwartz taught us. He didn't help us with where that they were bound or what DNA looked like. Just they're bound, because they're there in equal concentrations. So this gives us what we now refer to as Schwartz Rule. Right? As it says there, Schwartz noticed in his studies that DNA that there were proportional amounts, okay, of adenine and thymine, guanine and cytosine. So he deduced that A binds with T and G binds with C. Okay? Now, one little side note. A and T, they have to be equal. G and C have to be equal, but the proportion of the AT to the GC does not have to be equal. Okay, that's what makes our DNA special, and that's all part of our genetic code, which we'll deal with in another video. Enter Linus Pauling. All right, so Linus Pauling is actually the guy that first made a model of DNA. Only problem is he he made several mistakes. Like his was inside out. If you think of DNA like a twisted ladder, Linus Pauling had all the steps of the ladder on the outside like this, and then two poles that go up with nothing to hold them together, right? But he's the first guy to make a model, right? So it at least provided Watson and Crick a framework, so to speak, to start with. Then we get Rosalind Franklin and Maurice Wilkins. So Rosalind Franklin and Maurice Wilkins, they wanted to discover the shape of DNA just as bad as Watson and Crick. They were going about it a little bit differently than Watson and Crick. Rosalind Franklin and Maurice Wilkins were taking pictures of DNA using what was called at the time X-ray diffraction. So basically they would take DNA, they'd freeze it, and fire X-rays at it against a photographic film to try to get a picture. Okay. Unfortunately, they didn't really understand all of the safety issues when it comes to X-rays back in the 1950s, and Rosalind Franklin, the person who was taking most of the pictures, ended up contracting cancer from her work and died very, very young as a result. This is the picture that basically broke it wide open for Watson and Crick, but not Rosalind Franklin and Maurice Wilkins. And you might ask why. This is the picture that Rosalind Franklin took that, well, really helped Watson and Crick figure out that DNA was a double helix. How did Watson and Crick get it, and why did Rosalind Franklin and Maurice Wilkins not discover the structure for themselves? Well, it's a little bit of industrial espionage, if you will, in that, well, James Watson saw this on Rosalind Franklin's desk before she had a chance to look at it, and the rest is history. He was visiting her lab one day. The photographic films had been delivered. He goes through her stuff, and like, there it is. And he says, "Eureka, I found it." Ran down the hall to his buddy and his buddy Crick, and they, like, bada bing, bada boom, assemble a model, and there you go. And now they are given credit for discovering the double helix. Now, Watson and Crick were not evil people. I mean, they did give credit to Rosalind Franklin and Maurice Wilkins as contributors, but not as the discoverers of the helix. So this is where we end our story in terms of DNA from this video or these notes. The race for the double helix. So the idea in the race for the double helix is not a foot race. It's literally like a scientific investigation or a competition to be the first group or person to discover the shape of DNA. If you could do this back in the 1950s, I mean, you were pretty much guaranteed a Nobel Prize. And if you're not familiar with the Nobel Prize, on one hand, it's kind of cool because they do give you $1 million, but that's, I don't think, the main reason why people do it. Yeah, I think they're in it more for the bragging rights and the prestige than they're in it for, here's your $1 million, right? But at the end of the day, Watson and Crick were given the Nobel Prize because they were the ones who basically gave us the DNA double helix. Okay, so that's a brief history, just using some of the resources available to you through your textbook from chapter 12 regarding the discovery of DNA. We're going to continue to work on growing our knowledge of DNA in future videos. I hope you've learned a little bit about how all of this began through this video. I'm Mr. Kennedy, and I will see you next time.